in Dr. Jonathan Epstein's lab at the Perelman School of Medicine at UPenn. He studied molecular biology and history at George Fox University, graduating magna cum laude in 2016. His current research focuses on novel RNA delivery technologies to generate immunomedicine therapies in vivo, and is investigating how the cardiac mesenchyme interacts with the immune system following injury. Long-term, Joel is fascinated by the adaptive immune system and wants to leverage these powerful cells for human health. Outside of lab, he is an avid bird watcher and coffee drinker. Today, he'll be discussing his work on CAR T cells produced in vivo to treat cardiac injury.
Great, thank you, Alex. And it's nice to be talking with all of you today. I'm looking forward to the discussion, and hopefully have plenty of time for Q&A and idea sharing. I'm glad I don't have to go first, so that you have a wonderful introduction to what CARs are and the power and the breadth of their applicability. One of the things that we're really excited about in John's lab is to pull adaptive engineered therapy out of the oncology clinic and apply it to other uses. For us, that's starting with the heart. So when the heart gets injured, we get a buildup of what's called fibrosis. This is the extracellular matrix that is really stiffening the heart. There's a bunch of knock-on maladaptive responses, and it doesn't really matter what type of injury you give to the heart, whether it's acute or chronic, high blood pressure or genetic, stress-related. A lot of them end up at a similar phenotype where you get this buildup of fibrosis. Here it's stained in pink. We're looking at the left ventricle of injured mouse hearts, and a zoomed in view. You can really see this buildup of fibrosis. And this has independent and strong negative effects on heart function, and therefore the organism as a whole. It's a very big problem throughout the world, especially as humans live for longer amounts of time. And unfortunately, there's no real powerful therapeutics available. There are a few that have some promise, but haven't really translated to the clinic with all that great efficacy. So for us, we've used an engineered cell therapy. And as was previously discussed, and you probably already know, this depends entirely on a selective antigen to kill a certain type of cell that is maladaptive. And in cancer, that is a single protein that's expressed on the surface of the cancer cell that is very bad and exclusive to that tumor cell. So for us, we take advantage of the activation of fibroblasts. This is a process that is normal after injury, where a fibroblast on the left here is an active participant in tissue homeostasis, but of a certain molecular phenotype. And after a very complex and interconnected signaling cascade, we get a new cell state that is phenotypically and molecularly distinct, what we call an activated fibroblast. And this is what's responsible for the pathogenesis and pathologic features of cardiac fibrosis, and it likely holds true for many other fibrotic diseases, regardless of the tissue. What's really important for the talk today is that this activated fibroblast that's pathogenic is molecularly distinct from the resting homeostatic fibroblast. And through a fair bit of work, we have identified a single molecule called FAP, or fibroblast activation protein, that is exclusively expressed in those activated fibroblasts only after an injury and restricted to the injured areas. So what we're looking at here is a mouse heart. The top row is Mason's trichrome, so this time fibrosis is in blue, healthy tissue is in red, and you see there's not much injury, and correspondingly there's no green FAP expression. But after a pressure overload, so high blood pressure injury to the heart, you get these fibrotic lesions that mirror and match the FAP expression. This holds true in human hearts as well, not just in mice. So in a non-failing heart, we're still looking for FAP in green, this time counterstaining with cardiac troponin, which identifies healthy cardiomyocytes. And we do not see any FAP expressed in non-failing hearts, but in hypertrophic or dilated cardiomyopathies, we certainly see plenty, especially in the injured areas. So we have developed a CAR T that targets the FAP that is expressed only in these activated injury responsive fibroblasts. This is schematized below, and we heard a bunch about CARs just a few minutes ago, so I won't reiterate too much. Essentially, we get a targeted elimination of just these disease-causing fibroblasts. These are the same pictures you saw at the very beginning. So in an uninjured mouse, there's not much fibrosis. After pressure overload injury, which we drive with angiotensin II and phenylephrine administration, you get robust accumulation of fibrosis and a targeted elimination upon the administration of traditionally virally engineered or ex vivo manufactured FAP CAR T cells. So this was very exciting, but it does come with some problems because I just told you that fibroblast responses are essential across the body, irrespective of the injury type and across most or all of the major organs. So if you have an acute injury somewhere, you do not want a persistent CAR T to eliminate that necessary response to that acute injury. In the heart, that would be an acute myocardial infarction, as opposed to the chronic high blood pressure of which we've initially shown efficacy. And so, these virally ex vivo engineered cells are chronic. They persist in humans for decades, in mice for many weeks. And so we thought, hey, can we use mRNA? It's a really exciting time for the field of mRNA therapeutics. So we teamed up with Drew Weissman, who's an expert on RNA therapeutic development and vaccine development here at Penn. And together we worked out this really fascinating mechanism to deliver mRNA to T cells in circulation. And then lastly, I'll tell you about a really interesting finding that we found in the spleen that I really didn't have an explanation for until very recently. So together with Drew's lab, we identified this targeting schema that's reminiscent of the COVID-19 vaccine, where you have a lipid nanoparticle that packages mRNA. This has been modified, so it's stable and expresses incredibly well. The lipid nanoparticle is where the vaccines are administered. However, we go one step further and add onto the exterior shell of the lipid nanoparticle a targeting antibody. For us, this is CD5, which is a molecule that is normally expressed across all classes of T cells and is not required for effector function of the cytolytic T cell. So as you administer these LNPs, what we now refer to as targeted lipid nanoparticles, the antibody on the exterior shell of the LNP binds to the CD5 normally expressed on T cells, receptor-mediated endocytosis, escape, and translation, and we get a transient expression of the CAR protein. These now FAP CAR T killer T cells hone in on the heart, specifically the activated fibroblasts that are expressing FAP, whereby they eliminate them and the heart gets better. And I'll show you that data in the next couple of slides. What's really exciting is this has nothing to do with taking the cells outside of the body, and you inject this lipid nanoparticle directly into the bloodstream of the animal, and all of this schematized molecular biology occurs in circulation. So to prove first that we can get T cells to express RNA delivered through this targeted lipid nanoparticle scheme, I worked in vitro, taking isolated T cells, again, naturally expressing CD5, co-culturing them with these targeted lipid nanoparticles, waiting 48 hours and assessing for expression based on flow cytometry with various controls. And we can see here on the flow plot, we're staining for the FAP CAR on the x-axis, and you can see expression only in this red color, which has both the targeting CD5 antibody and the FAP CAR RNA. If we give a targeted antibody and expresses GFP, we don't see any expression in yellow. And notably, if we give a non-targeted, so IgG isotype control on the exterior of the LNP instead of the antibody, then we do not see any uptake by T cells and no expression of the mRNA cargo. So it really depends both on the cargo and on the targeting antibody on the exterior of the LNP. And we see that approximately 70 to 80% of T cells in culture take up and express some amount of the CAR. We next asked if these made functional T cells. And so we also performed killing assays with these, where we're taking target cells that are transiently expressing FAP and co-culturing them for 12 to 18 hours with the FAP-CAR expressing cells and watching them be killed as read out by a decrease in luciferase activity. And you can see again in the red salmon colored bar that there is exclusive activity, and it depends on both the targeting antibody and the RNA encoding the CAR. So that's fantastic and exciting, but we really wanted to do this in vivo in the animal. And so we moved to a model of high blood pressure. This is stimulated by an implanted osmotic mini pump. So this will continually deliver angiotensin II and phenylephrine, both of which independently drive high blood pressure. And this pump will be active for 4 weeks, so through the end of the experiment and beyond. And at 1 week, when we know fibrosis is established, FAP is robustly expressed on these activated fibroblasts, we will give a single dose of these targeted lipid nanoparticles, creating a single transient wave of functional FAP CAR T cells. And this single injection is fighting an uphill battle against the continued injury. So first, we wanted to make sure that the CAR was being expressed by T cells. So we looked 24, 48, and 7 days after injection of the targeted lipid nanoparticle. And here we're looking at a slightly different flow cytometry graph, but very similarly. This is all splenocytes that are alive from the spleen of injected animals. And we're looking at CD3, which marks all T cells, and then the FAP CAR on the y-axis, which is really exclusively expressed in T cells, both CD4 and CD8 subsets at 24 hours, and all other splenocytes that are not expressing CD3 really do not express very little FAP CAR. So we think it's fairly exclusive in terms of lymphocyte targeting to T cells. Notably, at 7 days, it is absolutely gone by protein, and we think the actual kinetics are closer to 4 days in vivo. So next we asked whether these cells get to the injured areas of the myocardium. Do they actually traffic? So first we're looking at, this is left ventricular myocardium. FAP expression is in this magenta color here. All 3 of these hearts have been injured, and in yellow is all T cells. After an injury, it's normal to recruit T cells. However, we see a real accumulation of T cells at the adjacent to the FAP expressing fibrotic lesions in the heart, only in the targeted LNP delivery, whereby the ice-type control or non-injected animals, we rarely see T cells. Then we co-stained directly for the CAR construct. This time this is in cyan, and we're still looking at the left ventricular myocardium with a confocal microscope and really only see expression on the surface of T cells in the targeted lipid nanoparticle condition. So next we looked by echocardiograph whether these hearts actually get better. This is now 2 weeks after the injection of the lipid nanoparticle, 3 weeks total. Echocardiograph, just in case you're not familiar, is an ultrasound picture of the heart in 2 dimensions plus time. And so this is a human example just because it's pretty easy. It's upside down, but this is how the images normally come out. So left ventricle here, and left atria, right atria, and right ventricle. So the left ventricle is what pumps oxygenated blood out to the body, and you're looking at the contraction or systole and the relaxation or diastole, and both are really important to the heart's function. So first we measured ejection fraction and global longitudinal strain. These are 2 metrics of systolic or contraction force. In gray bars, we have uninjured animals that are not treated. So this is what a wild type mouse looks like. After induction of the injury and no therapy, we have a strong reduction in terms of systolic function, and global longitudinal strain is the same pattern but opposite in magnitude. And a single injection of targeted lipid nanoparticles creates an almost perfect resolution. We see the same thing when we look at E over E prime, which is a metric of diastolic or relaxation function. The histology appears similarly, where we see a reduction in fibrosis of these hearts only in the treated targeted lipid nanoparticle injected animals. But what we do see, regardless of the method of creation of the FAP CAR T cells, whether they're ex vivo engineered or a Cree-driven diphtheria toxin receptor mediated ablation of activated fibroblasts or the targeted lipid nanoparticle, which is what I'm showing here, we see a latent or persistent perivascular fibrosis. This has been seen by many cardiologists for many decades, but it's never been parsed out from the other interstitial fibrosis, which is what you would see in the main portion of the myocardium. And so we're really curious to get a handle on what these cells are and what they're contributing to pathology and whether we can independently eliminate them with new upcoming CAR constructs in parallel to the FAP-CAR mediated ablation of the interstitial activated fibroblasts. But it's really interesting, and it really highlights the many different cell states of fibroblasts after an injury. They're not all homeostatic and they're not all activated. There are certain flavors of these cells. And it's really going to be interesting over the next couple of years to parse out these different types and see how they're independently contributing to pathology. So in transition, I first told you about the molecular biology that we've invented and shown to work. We get with a targeted lipid nanoparticle vaccine-like molecule, we get expression of the FAP-CAR that lasts about 2 to 3 days, certainly gone by 7 days. These FAP-CAR T cells traffic to the areas of injury in the heart where they lyse the cells and the myocardium recovers. Now, it was really interesting as we ended up seeing the antigen FAP from these activated fibroblasts ending up in the spleen and specifically in white pulp regions. We really had no explanation for this for a while until I went back and read the old literature from the 1990s on a process called Troglocytosis. This is a word that comes from the Greek meaning to gnaw or to nibble or to bite off. And it's a really fascinating process whereby T cells can uptake molecules from antigen presenting cells and keep them as their own without killing the presentation cell. This was originally identified in the late 90s, where a T cell here on the right and an antigen presenting cell on the left with MHC labeled with GFP, you see the immune synapse forming. And then over a short amount of time, there is small bright green puncta. This is the result of trogocytosis. So these are little membrane bits of the APC that have been plucked off and kept by the T cell. This has been studied further, although not extensively. And this is schematized here where the antigen presentation and the antigen receptor on the T cell are joined and then pulled off in small bits. And notably, not only does the antigen on the MHC get pulled out of the membrane, but a small bit of the antigen presentation cell membrane itself get taken up and kept by the T cell. What's interesting is that this is not exclusive to T cells. It's not exclusive to APCs. It happens in amoebas all the way through other cells in the body. And it has been identified in vitro and in the tumor in CAR T cell therapy, but it has not been extensively studied. So first, to show you that this process can happen with the FAP-CAR and a FAP-expressing target cell in vitro. So this is schematized here where we have RFP directly linked to FAP, the intracellular fragment of FAP, whereby the rest of FAP is on the exterior leaflet of the plasma membrane. And then we add FAP-CAR expressing T cells and then watch over time as the RFP is transferred to the T cells. And hopefully this movie will play okay. I'll have the relevant screenshots in the next slide. So we have RFP expressing target cells. You can see an immune synapse forming, and then puncta within the cell. And you'll see a second CAR T cell coming on the right here, an immune synapse, and then puncta within that cell. This is FAP being extracted and retained within FAP CAR T cells. So here's the screenshots of 5 minutes. Nothing has happened yet. An immune synapse forms, and then you can see individual puncta while the second T cell is there, and then puncta within both. So what's really interesting is when you're looking now at the white pulp regions of the spleen, so this is where T cells collect, that's what's dashed in these white outlines here. And only after an injury in the heart and an administration of FAP CAR T cells do you see these white puncta of FAP, the antigen from activated fibroblasts. held within T cells in the heart. So if you don't injure animals and you give them wild type T cells, you don't see anything. If you injure the animals with angiotensin phenylephrine, but you don't give them wild type T cells, you don't see anything. So the injury does not induce local FAP. It's really an induction of FAP in the heart. And then only after you have both the injury in the heart and the circulating CAR positive T cells do you end up seeing FAP exclusively in the white pulp regions. If we zoom in and used a GFP labeled version of the FAP CAR, this is again the white pulp region of the spleen. You can see a bit broader of a view, and if you zoom in a single Z slice, the pink here, or magenta, is FAP coming from activated fibroblasts in the heart, ending up within T cells that were engineered in the spleen. We think it's a really interesting observation and needs a lot more work to understand what it means. Notably, this also occurs in the targeted lipid nanoparticle, although it's a little bit harder to find these Troglocytosis T cells there because they are quite transient. So just in conclusion, I'm happy to answer some questions. I hope I've convinced you that we can make transient active FAP CAR T cells in vivo using a targeted lipid nanoparticle product. This product is mass manufacturable, and importantly, it's very dosable. So that as you need additional doses or your heart is re-injured over time, hopefully you'll be able to come back, get repeat doses of therapeutic, making transient waves of anti-fibroblast CAR T cells, and then the T cells will go back to normal, and you're able to respond to acute injuries as normal. We also show that FAP CARs are able to trogocytose FAP from activated fibroblasts and collect in the splenic white pulp, although we're not quite sure what that means yet. And as I'm sure you can imagine, there are hundreds of open questions that are all interesting to me and hopefully interesting to some other people as well. So how does the recruiting excess T cells to the injured myocardium impact the local inflammatory environment? Is there any impact of this trogocytotic process? What happens to the T cells after the CAR is gone? Do they have any memory of targeting? Is there any epitope spreading? Is there any residual memory of this? There's certainly more fibrotic diseases than heart failure, including large animal models before we move into clinical trials. And then the perivascular fibroblasts that I highlighted very briefly, they probably are contributing independently. And then lastly, there's a whole independent field of positron emission tomography imaging field that has developed FAP imaging probes where you can see FAP expressed anywhere in the body, both in cancer-associated fibroblasts, but also in heart injuries of various kinds. And so we're really excited to pair up with some of these imaging colleagues of ours and to identify potential patients and screen them before and after therapy for efficacy and off-target effects. So with that, I would like to thank my mentor, Jonathan Epstein, and the rest of our lab, specifically Haig Agajanian, Lee, Pedro, Swapnil, and Drew's lab, including Hamide, who's our own PI now, Isfan and Amir, who really were essential in creating the product that was essential to our studies. Our funding agencies are broad, especially the NIH, and I'd be happy to answer any questions. Thank you.
Thank you, Joel. Great talk. Very interesting delivery system. So the first question is, how long does the CD5 LNP remain in the blood after one injection? And specifically, what are the repercussions of the CD5 lipid nanoparticle in the thymus, as CD5 is expressed on thymocytes and regulates positive and negative selection?
Oh, all right, so I might have frozen for the very last part of the second question, but I'll try my best, so remind me if I missed something. So the targeted lipid nanoparticle in circulation does not persist for long, probably on the order of tens of minutes to maybe an hour or two before it falls apart. And so we think that the delivery of mRNA happens very quickly. The mRNA is then stable and expressed for approximately 3 days, 4 days, before degrading naturally. So we don't think there's any overt concern for systemic toxicity due to the LNP itself, although that is something that we're actively working on iterating many different lipids, many different compositions, and many different styles of LNPs to try to minimize some of those off-target lipid nanoparticle-based cytokine release. It's different than the cytokine release syndrome that Louis talked about earlier, but it is a potent adjuvant on its own, which is great for vaccines, less important for us. In terms of CD5 expression in the thymus, we're not aware of any negative impact of delivery of the FAP CAR to these cells, but it is certainly something we will look into.
Thank you. This one is from Louis. In addition to direct killing of activated fibroblasts, are there any additional payloads that could be expressed within the engineered T cells and presumably localized locally at the heart to promote resolution of fibrosis?
Yeah, certainly. So, I presented one application of a targeted LNP. The targeting antibody certainly does not need to be CD5. The payload does not need to be a CAR at all. What's tricky is trying to get temporal expression in the heart as these cells traffic through very quickly. It only takes a few minutes or seconds for blood to circulate through the whole system, especially in a mouse. So it's a bit hard to get localized delivery in an organ, although we're trying to brainstorm some ideas. So we'd love to hear if you have thoughts on that. One interesting use of modified RNA in LNPs is AstraZeneca is working on a delivery of VEGF-C to stimulate angiogenesis after an injured heart. So after a myocardial infarction, so an acute injury, a few days after that event, then they'll basically directly dose in some LNPs into the area that contain angiogenic factors to be expressed. So something like that is certainly possible. What we're hoping happens with the antifibrotic T cell is it clears out some of the most maladaptive cells so that the rest of the complicated milieu of cells are able to recover and kind of tip the balance away from a fibrotic response towards a pro-regenerative response based on other endogenous factors.
Okay, we have another one. Could this methodology be implemented for treating idiopathic pulmonary fibrosis?
IPF is something we're very interested in. It's the mouse models for IPF are a bit confusing in terms that they don't necessarily directly replicate the human disease. And so we're trying to evaluate which mouse models to test this in to get, we don't want to put the work into something that doesn't replicate the human, because if we get a negative result from an animal model, just because it's the incorrect model, doesn't mean it won't work in a human. So it's a fantastic idea, or something we're very intrigued with. We're just not quite ready to test it in like a bleomycin model.
I have a question specific to the troglocytosis. I know very little about troglocytosis, but I was wondering if potentially changing the affinity of the scFv or even changing the transmembrane domain, would that change the outcome of whether troglocytosis occurs or not, or is it more on the antigen end of things?
I don't know for sure. What I do know is that that CARs seem to be particularly good at stimulating troglocytosis. So there's an eLife paper from a few years ago where somebody not only transduced T cells to express the CAR and they see troglocytosis in vitro, but they also transiently overexpress the CAR in fibroblasts, in 3T3s. And those cells are able to trogocytose FAP out of target, I think they were Charcot cells. So it really seems to be strongly stimulated by CARs. They used two different CARs. We used the third with FAP. And so I don't know. I just don't know what it means. Some people argue that it helps tumor cells downregulate the epitope that's being targeted, the antigen. because it's pulling them out without killing the target cell. But the data for those is confusing and somewhat contradictory, so I can't really make any firm claim, although what I do know is that the CAR construct itself is exceptionally good at trogocytosing its antigen out of a target cell, regardless of what the host cell is.
What about using an engager rather than a CAR? Would that prevent trogocytosis?
It's unknown. We haven't tested them, and we know a FAP engager exists, and we know the person that would do the experiment, but we don't know of any data. So what I strongly suspect happened is that it was attempted and it hasn't proven to work, because engagers are not as potent as the CAR T is. And it's much more likely that the recruitment into the heart of these cytolytic cells requires the CAR and not just an engager.
Okay. We have one last question. This is kind of broad, so this could go to either of our speakers.
Sure.
How was it possible to reproduce the model or in animal models, or to infuse the CAR T cells into animal models? What requirements do you need as a laboratory?
Right, so I'll take a stab, and then Loay, you're welcome to chime in. For us, one of the goals, and I didn't really make this explicit, was that this product is fairly mass manufacturable, not only for clinic use, but for research as well. And so we don't need any form of ex vivo manufacturing capabilities. Really, it's just it's injection of the targeted LNP, which admittedly, it's a bit hard to get LNPs made right now because all of the manufacturing is going towards vaccines. But it's getting easier, which is good. In terms of isolating and ex vivo activating T cells and engineering them with virus, it takes some practice and it takes some money. Other than that, basic reagents that you have in your lab should suffice.
Yeah, I think I guess I can answer part of that as well, but I think typically for producing CAR T cells to be injected in mice, there's kind of one of two ways to do it. You could either do an immunocompromised mice where you would be using human T cells, and you would have to be using PBMCs. So that would be taken from human donors. You isolate the T cells, and then you can retrovirally or lentivirally infect them in vitro. Our lab usually does about 7 days where we grow them up in vitro, run flow to make sure the CAR is being expressed, and then we can inject them intravenously into the mouse. With an immunocompetent model, you would have to actually take T cells from the mouse, and you get that usually by harvesting spleens and isolating the T cells. Either way, it is quite a bit of work, and it usually takes 7 to 10 days to produce the T cells before they are injected in vivo. Great, I think that's it for questions.
So yeah, thank you very much, Alex. Thank you, Joel. Thank you, Loay, for this great presentation and this great discussion. So anyone in the audience, if you're interested in open book science, I just put it in the chat box, our website and a Google form that if you could, propose a type of symposium, a specific, but a particular topic you're interested in having in open book science, we are really happy to hear you. Thank you, Alex, and thank you, Joel, and thank you, Loay. Thanks for creating this great science, and we will hear a lot about you in the upcoming years. Thank you so much. Thanks, Alex.
Thank you.
Bye.
Thank you. Bye.
Bye.